Oh, perfect. Great. Thank you. Well, thank you everyone for joining us today. It's my pleasure to be speaking with you all about the CAMH BrainHealth Databank initiative. We're really focused on digital health data as well as AI for personalized care. And I hope today that I will give you an idea of sort of what we're able to do today to support enhance clinical decision-making in the clinic and what we're hoping to be able to do tomorrow. Hopefully this ties back to some of the themes that you've been hearing earlier and talks you've been hearing earlier in the week and last week as well, with respect to some of the challenges in the clinic that Victor and Brett spoke to you about, some of the work of the other scientists in the Krembil Center for Neuroinformatics, and some of the themes that Abby touched on today. Just by way of introduction, my background is in molecular genetics. I was doing preclinical work looking at molecular mechanisms of mental illness, but I quickly realized that I was very interested and keen to do something that was more translational in nature. And since that time in grad school, I worked at a number of different organizations, including the Ontario Brain Institute, as well as CAMBINE, which is a national biomarker depression research and education network. And so there I had the opportunity to work with a portfolio of multimodal clinical research studies. But again, I was really and I was sitting in the clinic, but I didn't really feel directly involved in the clinic in the way that I am here today with this work. And so I hope that comes across in the presentation. Oops, sorry. So just to start with, this work that I'm going to be talking about today is really not possible without collaboration with various different experts, as well as different technology tools, and of course, computation. So our goal is really to focus in on the citizen patient and really improve mental health outcomes. And so, but to do that, we feel that it's very important to work closely with the clinic and the clinicians, directly with them. Obviously bringing in together into that mix are researchers and clinical scientists. And also not shown here, but really important as well, engaging with patients and families themselves. Of course, privacy, legal, ethics, data engineers, and some of those pieces that Abby had spoken about. And the tools themselves, the digital tools themselves. So we're fortunate here to have a fully electronic health record system. Of course, we're very interested in the collection of multimodal data, so everything from genetics to brain imaging and wearables. And we have something called the Neuroinformatics platform, which really provides open source tools to enable us to collect these different types of data in a standardized fashion. And of course, the computation. I think the sound is still turned on.
Yeah, okay. Well, I solved it.
Oh, awesome.
Sorry to interrupt. So keep going. Keep going.
No
Great. Sorry, Joanna.
Great. So really our goal here is to create this sort of continuous learning health system in which we can use data from care, integrate it with research studies, increase our knowledge, and also then apply that knowledge back into the clinic. And some of the key pieces at CAMH and our approach that allow us to do that is that we have already set up measurement-based care. And so you'll hear a little bit more about this, but really it's the structured and standardized collection of measures along a patient's treatment trajectory. And in this case, in mental health as it stands right now, it's largely dependent on self-report assessments, as you've heard, which have their challenges. The other piece, as I mentioned already, is really important to sort of the work that we're doing is having an electronic health record system. So all of the clinical data is stored here. It's a source of truth. We're able to extract this data and use it at point of care, but also to carry out various different research studies and looking at development of AI and machine learning prediction tools, et cetera. The last piece that I mentioned already is our Neuroinformatics research platform. And for us, this is really important because it allows us to capture the multimodal data types that we're interested in. Am I sharing my slides full screen or did the view change?
It's like a half screen, half and half. I think you could still make it focused.
Okay.
Abby might have to do that. Oh, there we go. Oh, now Abhi is full screen.
Okay.
That's good.
Perfect. Sorry for the technical difficulties. So just briefly to go over some of the considerations, I think, which are probably important for this group in particular, when developing AI machine learning modeling tools and technology to advance personalized treatment. Just have sort of three themes here that hopefully will come up throughout this presentation and how we're addressing them. Of course, it's really important to have a sustained collection of high quality standardized clinical data. And so without this sort of standardization and consistent quality, it will be difficult to have reliable clinical decision support that can be used in the clinic. And so we have a couple ways in which we're supporting the collection of data as well as the standardization, which I'll speak to in a few moments. We also here are now collecting data that's reflective of the real world patient population, and for us, the patient population being that of CAMH. And so this is important in terms of facilitating translation back into the clinical practice, which is one of our key goals. And, it's also important, as you've heard about, to consider caveats. So, are there limitations in terms of the representativeness of the population that you're looking at? Especially if you're going to extend to other populations outside the one that you're working with. The last one too, of course, is access to this so-called big data. So, how do you access this rich longitudinal multi-scale treatment patient-directed data? We do want to have a robust governance such that this data can be reused to answer many questions. And in order for the data to be reused, of course, we need to really ensure the longevity of the data. So just to orient you to the work of the BrainHealth Databank, we've broken it down into 4 key objectives. The first being digital support for measurement-based care. And sorry, I should say all throughout these 4 key objectives, we've really worked on establishing sort of new technology or infrastructure to enhance what we already have here at CAMH in order to operationalize each of these aspects and really enhance and support our capability to move forward with them. So I'll just quickly give a highlight of that, but then I'll describe how we're sort of applying them currently in our clinical setting today, and again, like what we're setting ourselves up to do tomorrow. So for the digital support for measurement-based care, we have technology in place to support the scalable collection of electronic measurement-based care. So what this means is pre-pandemic, patients would come in, use a tablet in the clinic to collect their self-report assessments. With the pandemic, we transitioned to virtual care, and we're now able to send folks links to these surveys that they complete still ahead of their appointment. Results are automatically calculated, and the scores are provided to their clinicians ahead of their appointments and used then to inform treatment decisions. Of course, we know that there's some challenges with these self-report measures in terms of variability, recall issues. We talked about them being sort of subjective in nature. So we're very interested to layer in additional research measures into these treatment pathways, so into these standardized care pathways. And of course, we're interested in genetics and molecular data. So on that note, we've launched a biobank here at CAMH to support with the standardized collection and processing analysis of these biosamples that we collect, as well as Actigraph's service device rental. So, pathways that are interested in layering in, looking at daily sleep and activity, are able to integrate these devices into the research. Of course, the idea is to then use this data and really feed it back into the clinic to enhance how personalized care is delivered, and as we talked about, really augment the decision-making process. So by no means replacing the work of the clinicians and conversations and joint decision-making with the patients themselves in terms of treatment decisions, but rather providing additional tools and information, data, evidence to inform the decision-making. And so with that, we have been working on infrastructure to support real-time data flow for dashboards, which I'll speak to, as well as AI pipelines to draw new insights from data that can be used and fed back into the clinic to, again, inform decision-making. The last piece is open science for discovery and innovation. So we talked about how we want to be able to reuse this data for secondary and tertiary purposes, as well as to grow our knowledge base. And so we've definitely implemented ways to be able to search and query the data, because you can only ask questions if you know what's there. And of course, the FAIR principles, which we'll speak to later, as interoperability and ensuring data longevity is important for both reuse of the clinical and research data we're accumulating. So for digital measurement-based care, briefly mentioned that the idea behind measurement-based care is to monitor changes in symptoms over time. We're heavily reliant currently on self-report assessments, and so the idea is that patients come in, they complete different assessments, not only at baseline, but throughout their treatment journey here at CAMH, and of course at discharge. Whilst this seems like many should be doing this, and I will just say that in psychiatry, it's not necessarily common to all practice. You often see it used a little bit more in things like psychotherapy, as they're basing their therapy around these changes in symptoms. But regardless of that, it's set up now in such a way that makes it takes away the burden from their clinician. So there's not, it's not clicked on a paper, math doesn't have to be done. It's sort of hands off from the clinician's perspective, in that they were able to receive a report with these summary scores and how they've changed over time that they can use to inform treatment decisions. So I'm mentioning how this helps the clinic because I think this is also an important aspect if we're interested in collecting high quality data. It's important to also be working with the clinic operations who support often scheduling of appointments, understanding when people are lost to follow up. As you'll see, the data is very heterogeneous in nature. Some people may come from 1 visit, 2 visits, but in terms of understanding the full trajectory, it's helpful to know, did they get better, and that not why and that's maybe perhaps the reason that they didn't come back. So really working with everybody who's involved in the data collection process to ensure that we continue to improve our data quality is very important. And one way to do that is to make sure that they're also getting something back from the data that helps them to do their job. And so, I'll touch on what that could look like. Sorry, just due to time, I don't have time to show, but basically we are building a dashboard to help people see the flow of patients through the clinic and utilize that to make corrections to the electronic health record to show if they've been discharged or reasons for loss to follow up. So I do just want to give you an idea of how wide this is spread. So we started with one pathway, depression pathway, but we've really expanded to 15 different care pathways. So there's ones for alcohol use disorder, bipolar disorder, opioid use as well, and addictions. We have over 7,500 patients enrolled and across some different clinics, and of course, well over 125 clinicians involved. So we mentioned that, the collection of measurement-based care and these self-report measures do have their limitations. So our next step is, once we have this process really well established, is to layer in additional research measures. We're very interested in biobank samples for molecular and genetic data, as well as the actigraph data for daily activity and sleep. And so just to give you an example of a study that we're looking to launch later this summer, hopefully in August, we've taken our depression treatment pathway, which already, as you can see, in this top box has a number of different measures that are collected over time. So the patient self-report, from the physician, the treatment plan. Some people are set for blood work for clinical purposes. And also everyone has the option as well to participate in group psychotherapy. And the reason why I mention that is that this group psychotherapy also has a stream of self-report measures that can be used to assess how people are responding to treatment. So our idea here to augment the measures that we have available is to increase the number of self-reports to give a more holistic picture for when folks are not in the clinic. I will mention that one of the self-reports that we have is inviting patients to identify outcomes that they're interested in improving. So sometimes, the clinical assessments are sort of predetermined by them, and there might be other outcomes in terms of functional outcomes that they're interested in seeing improvement in over time. So really important to have the patients engaged in their mental health journey. Additional clinician-related rated scales to supplement some of the measures, and of course, these more objective biobank and actigraph measures. And so while we're collecting all this data, then becomes quickly, you can see that there's a large amount of data to be potentially fed back to the clinic to help with decision making. And so where we started, again, this is what we can do today, is that we've taken the data that we're currently collecting through the electronic health record and presented it back in a dashboard format. This has been co-developed with some of the depression physicians to show at a glance a patient's treatment trajectory. So I didn't mention this here, but in this instance, this pathway does have a medication algorithm that's based on the depression severity score. So you either sort of stay on the same medication, or you're adjusted to a different dose, or if you're not responding to a different medication, and eventually escalating into brain stimulation as well. It flags the side effects. It shows here, this is sort of the key part in which it shows your depression severity, anxiety, changes in weight if weight is collected. We know antidepressants often result in weight fluctuation over your medication treatment. So you can see at a glance, if you're on, this medication, these medications here, your depression severity and anxiety seems to be worsening, whereas the switch in medications here seems to show improvement in your medication, as well as flagging the suicidality risk, which right now is currently just a single question in the depression severity scale. And so, of course, there's room for improvement to include new tools to be able to augment the decision-making process. And I've just given a number of different examples here. Obviously, this won't be the way to present it back to the clinic as it quickly becomes very busy. But for genetics, you could see returning a polygenetic risk factor, looking at pharmacogenetics to see if you're a fast metabolizer from one of the drugs, developing a response prediction tool. So really, from different types of data that's collected about you, do we think that you're going to respond to citrulline or not? Improving prediction for suicidality risk and monitoring, potentially using cognitive games that Andrea had talked to you about earlier in the week. For sleep and activity from the actigraph, even sort of boiling down that data that's been collected, say, over a month while you weren't in the clinic, or potentially longer, to be able to use this data to inform treatment decisions as opposed to relying on, how well did you sleep? Oftentimes clinicians will say, patients sometimes say they don't sleep at all. Well, is that really possible that you didn't sleep at all for a month, or what does your sleep look like? So how can we just provide descriptive data back? Another aspect is also using natural language processing. Currently, there's a lot of rich information in progress notes, but there really isn't the ability to search, and also progress notes can be quite long and lengthy, and when you have 10 or 15 minutes with a patient, it's really difficult to go through all of this information. So can we develop a way to summarize some of the key information, or provide a search function, making it easier to access? And so as these tools, of course, are being developed, it's really important to acknowledge the potential limitations, as we spoke about the diversity of the sample. And one way in which we're looking at including this is developing a health equity dashboard. So we're engaging folks from across the hospital, both on the clinical side and research side, and looking to have a tool to show you the health equity, the representative of who's, for example, in the depression pathway, so that you can use this to design your study, for example, or perhaps if from a clinical perspective, look at different ways of enrollment to increase the number of, to increase the diversity or representativeness of the population. We're also able to compare the representativeness to the Greater Toronto Area by using census data, just as some examples. Another important aspect, of course, is really engaging patient and families in their treatment journey, and also to make them more aware about their symptoms of their mental illness. And so at the same time as we're developing these tools that are clinician-facing and facing the clinic, we're working with patients and families to co-design sort of an equivalent or a similar dashboard that really shows their clinical journey. And so here's just an image of a very early on prototype, which we hope to build out more of, and be able to have patients use this to further engage in their mental health journey. This one here is just sort of showing mock-up of some of their clinical treatment data, but the same thing goes with the study that we're running. We have plans to co-design a dashboard to be able to return to them information about their daily activity and sleep. So Last key objective here is the open science for discovery and innovation aspect. We have collected a large amount of data so far on the clinical side that we think is standardized and of high quality. And it's being used, as you can see, for a number of different quality improvement and research projects, developing new tools, as well as natural language processing. But just to give you a sense of how much data there is, and of course, to touch on the standardization, which was mentioned before. So whilst, we have 7 different clinics participating, and of course, many different conditions being treated, there are some scales that are being used and deployed in a standardized fashion across. So you can see the depression scale being the most common, anxiety is being collected across as well, disability, and as appropriate, a scale for alcohol use. And so we're able to start to use this data to get at some of the heterogeneity that we're seeing. And I just thought I would include this picture to give you a sense of how heterogeneous the PHQ-9 or depression severity trajectories are from the depression patient population that we're looking at. And so you can see there's a number of different variables from how long people stay on the program to what their scores are. But of course, we're working with Dan and Trudra and others to start to develop subtyping and start to develop ways to predict who is responding to treatment. And so that's just one example, but of course we're able to and would like to ensure that the data from the clinic and the data from these research studies are available to not only researchers at CAMH, but researchers around the world for asking secondary research questions. And so the idea is to build up this brain health atlas that spans all these different scales and across the lifespan, to have it, of course, searchable. And the way that we hope to do that is to develop a cohort explorer so people can come in and see what data is available to inform the design of their research questions and studies, along with the robust governance. So I don't have time to go into detail with that, but that's really working with privacy, ethics, legal, patients and families themselves to really design a robust consent process to allow for this reuse of data. And I should say that it's not only data that's collected from research, but of course we want to include data that is collected from the clinical side as well, anonymize that and make it available for reuse. And so important to that, of course, are the FAIR principles to ensure the longevity of the data. I mentioned that it needs to be findable, so our way of addressing that is by providing a cohort explorer to allow people to search what's available. Having it accessible, we will have a data steward role in our governance as a resource to help generate the approved data sets once they've been approved by RED and various different governing bodies. We do want the data to be interoperable, and so we've adopted standard ontologies, one being FHIR. And of course, the data to be reusable. So there needs to be standardized data documentation to describe how the data was collected, the context in which it was collected, very important to understand if there were changes along the way, and really providing that sort of full picture for the data to be able to be reused. So just to finish off with, I hope I've shown to you today that we're really well on our way to developing this continuous learning health system. And we've touched on a number of different aspects that really bring and integrate care and research on an ongoing basis. And just lastly, to thank the whole entire CAMH-wide Brain Health Databank team. As one can imagine, this is definitely not the work of any one person, but of course, of a number of groups, from the clinicians to operations to scientists to data engineers, software engineers, privacy, legal, ethics, patients and families themselves to make this possible. And with that, let's stop for questions.
Thanks, Joanna. I don't see any questions. I've been looking at the board, but people can continue to put questions into the question zone or in the chat, and we'll get to it after. But since everyone here passed my test at 10:30 and stayed on for the approximately 10 to 15 minutes of faces and silence, you're all in the elite of summer school attendees. I have no doubt that there will be very interesting questions asked based on Joanna's presentation, BrainHealth Databank, and what's about to come. But I'll just dive in now to the second part of this session, and it's okay that we are, going to have to go at a bit faster pace to get through it, because this session was designed to give more of an overview and smorgasbord of available data resources. Joanna just talked about a hospital-based resource. It's the melding research and care. It exists behind a closed door of CAMH at the moment, aside from the open science and sharing initiatives she talked about. I'm going to talk about data that's out there now that you can access. That you may be able to access very large databases. And if you get anything from this, you might be able to just pick off one or two resources you didn't know about before, and that would be a win. So it's okay. We're not going to miss any major scientific value here, at least on my end, with the time crunch. So I'm going to I'm sharing the screen, but it's not popping up. Abhi is still pulling the strings at the moment. So there we go. Okay, so that looks good. And please, anyone, let me know if something goes wrong, because contrary to what you might expect, we did practice this. This was planned. So sometimes you just never know what's going to happen. So anyway, this is the purpose. How do we facilitate large-scale integrative research? We just heard about Joanna from our local initiative, and I'll actually segue into this later. It's a bit of a temporal anomaly, but everyone here is very smart. So data, you need data for this type of research. We need lots of data. It's not long ago, 15 years ago or less, sample sizes for psychiatry, when we're looking at data types that we've been talking about over the last week or so, whether it's EEG, functional MRI, structural MRI, genetics, genomics, we're talking about usually dozens of people. It was very difficult to recruit people who had clinical diagnoses and were willing to go through with certain types of assessments, obviously ranging from invasiveness of a blood test to being in a tiny scanner. Your head is in a little donut, and it's very hard to see anything, very stressful experience. So that time has kind of come and gone. I mean, now we have hundreds up to tens of thousands and hundreds of thousands and even into the millions of individuals to study across populations, across clinical diagnoses, and across data modalities. I'm going to share with you an overview of some of those data sets, how we access them. Talking about data access, a lot of you will have worked with data. Actually, I expect everyone on this call has worked with some type of research data. It will have come through either someone, you work in a lab and your PI generates this data, like on a preclinical model, or they have their own sample of individuals that they've collected over years, and they give you the data and they say, don't tell anyone, don't show anyone, it's our data, we publish on it. Again, that's from a bit of a bygone era. It still happens. When we share data, we're talking usually about these 4 modalities of sharing. This is an excellent recently published article that kind of outlines how data sharing happens across. This is specific. They're talking about genomics. The data are based on genomics, but it's relevant to all forms of biomedical data that we use in psychiatry. And they talk about not just kind of how it works, but how funders look at it. How do funders, government agencies and various donors, make sense out of this financially? How do you get a return on investment at the public health level, or just from an efficiency standpoint in research by sharing data? And the data sets that I'm going to talk about today mostly follow this mode. There's only a couple exceptions to this, and this is controlled access sharing. So this is where the sharer, the PI, who in the past might have been very greedy and not share their data, agrees to put their data in a centralized repository, and there's a data access committee that is kind of separate from the sharer, but it involves the sharer actually, and the requesters will go to the repository and kind of fulfill some requirements. You have to demonstrate you're from a bona fide research institute. You have to give a proposal of what you're going to do with the data. You have to say that only some people will have access. It'll be secure. It'll be confidential. All of that, and then you get the data approved or not. That's the model that a lot of research is shared on right now. So I'm going to start with multi-omics data. It's a good place to start, namely because it was one of the first kind of large-scale data sharing initiatives. Was kind of started with these genetic databases shared by the National Center for Biotech Information, Biotechnology, NCBI, which is NIH funded. And this is kind of, it started at like year 2000 when the first sequences of the human genome and then, you know, other animals began to be sequenced. The actual letter sequences of organisms were shared. The kind of like, you know, just genetic sequence data. It's not paired with anything, and it's not a large number of people. It's just like a reference genome. It's data. It's not data we think of when we think of population studies, many people and paired data. But it's grown into different databases. So NCBI runs the gene expression omnibus. So if you're looking for gene expression experiments, so you know, different levels, we heard about this last Wednesday. If you want to understand how genes are expressed in different tissues, in people with various diagnoses or in different age groups or biological sex, you can look at Gene Expression Omnibus. The data set I want to mention is dbGaP. It's not a database. dbGaP is a resource for, so it's a database of genotypes and phenotypes. You see, this is an integrational database. So you can access genetic data and paired phenotype data for, you know, they say there's 1,200 studies. Some of them only have a few subjects. Some of them have many thousands of subjects. The Alzheimer's disease sequencing project. I think PNC, I think the Philadelphia Neurodevelopmental Cohort, which I'll mention, is accessed through dbGaP. Studies from around the world make their data available to researchers through that model I showed in dbGaP. It's the intermediate, and it's one of the older ones, and it's beginning to show its age, and I'll tell you what I mean. Synapse is another one, multi-omics. Actually, Abi used to work for Synapse. He was working on the Synapse team. And again, it's a central repository. You have studies that collect data and manage it, even, you know, in their own servers at their own hospitals or universities, and then give the data to Synapse to manage and organize. And it's quite nicely organized. You can see this is an example from a study, the religious order study. Actually, sorry, this isn't the religious order study. This is ACT. This is a similar study. It's an aging study that includes multi-omics data. And you can see, you can access an individual study. You can select an organism, because some of these studies make data available for you know, in vitro cell lines that they've gathered on the same subject. So we want the human data. We want to look at, you know, you can select metadata, proteomic data. It's all hierarchically organized, and it's quite conveniently done. You need access privileges, again, so it's not just because the data exists in one repo and you can look and pick and choose. Every time you want access to a particular data set, for example, the Common Mind Consortium, which is post-mortem gene expression data from people with psychiatric illnesses. And you have to demonstrate to the people who own that data, right, who generated it, that you're going to do something worthwhile with it. So it's centralized, but also split. You have to apply for these things in tandem. But Synapse has a uniform structure that makes it very easy to navigate. This is another example of kind of what it looks like. It's very pretty. There are lots of dashboards. This is from the Accelerating Medicines project for Alzheimer's disease, and you can kind of see a bunch of offshoot studies that are all related to Alzheimer's disease and part of this single grant, and they've made data available for gene expression, proteomics, metabolomics, and you get the idea. One thing that Synapse does that other data sharing tools don't actually do very well is they have a people section. They have like a wiki that runs in parallel with the data, so you end up with a functional situation where the data sets exist, you click on them, you look at information, you don't really know what's happening, you might have a question about missing data or what a field means, or you say, well, you've made a spreadsheet available and it says, you know, age, and the ages are all grouped in some way, and you want to understand why they were grouped that way. You can ask in the wiki, you know, you say, hey, why is this done? And people who have actually imported the data and communicate with the individuals on the study side can answer those questions and give you the answers you want. So that's a really nice, it's a nice platform. This is the Psychiatric Genomics Consortium. It's not really a data repository, as I kind of said these would be. This is a reference. It's a resource. It's not really a resource. So it's a consortium and a resource. The consortium is just a group of people, and you know, this is an international consortium, and they cover multiple diseases and have multiple working groups. This was kind of the first group of people who said, basically after, you know, the sequencing of the human genome several years later, said, let's start pooling all of our genetic data sets for these, you know, difficult to recruit patient populations, and start performing meta-analyses, so we can get the sample size up, so we can boost our statistical power, almost exclusively at the time for the purpose of running GWAS, these genome-wide association studies. So now it's grown. They've got all these working groups: schizophrenia, bipolar disorder, major depressive disorder. Now this PGC sample is kind of one of several that are, kind of cover even broader population-based data sets, as I mentioned. I've mentioned in the past UK Biobank, 23andMe, when I talked a bit on Wednesday about GWAS. But this is it's something you need to be aware of if you want to do genomics research in psychiatric illness. There's also the NIMH repository and genomics resource, which is it's pretty updated. There's 221 studies, but they actually have more of a synapse. It's almost like synapse in that they cover a lot of information, ranging from clinical information, genomic information, and like family-based information. It's got a much broader array of samples. It's not like PGC, although they do have family data in PGC. It's not like a lot of repositories that don't allow you to kind of go in deeper and critically make biosample requests for these individual studies. So if you wanted to apply your own assay or something to some tissue from one of these data sets, you can go into this genomics resource and request biosamples. You can do that for a lot of other databases. You often have to do that through a specific organizational portal. And I wanted to mention this kind of commentary, which was recently published, which is the broken promise that undermines human genome research. So these databases kind of started with NCBI and are now quite prominent. They were all kind of born out of this idea of data sharing, which isn't unique to genomics. It's in neuroimaging as well, all biomedical research. And the contention is that right now, and if you've been working with these data resources, you'll probably know what I mean. It's pretty unfulfilled. So this data is supposed to be open, but you'd be surprised at how difficult it can actually be to get access to a lot of this data. And they specifically point out, dbGaP. They kind of throw it under the bus because it's an aging platform, and it's really hard sometimes to just organize your documents. And it's like Canadian Common CV. If you're working on that, they are coming out with a new version of Canadian Common CV. But right now, NCBI is thinking if they should cut their losses with dbGaP or kind of fund a dbGaP version 2.0. We're at this stage where some studies that I'm about to show you are kind of picking up the slack, moving into the next generation of data sharing and data availability and federated data analysis. So as it stands, if you're accessing these data sets, dealing with data use agreements, trying to figure out what's needed and what the requirements are, it is frustrating. It's not just you. You're not stupid, and people aren't, well, not everyone is incompetent, but it's difficult, and it's not a good system. So keep that in mind and persevere, and know that it's really not you. Results browsers. This is something that's kind of come out of the hesitancy of a lot of organizations and labs and consortia to share raw data, and also fear in the population about sharing genomes and identifiable molecular data. Why make our genotypes and our phenotypes available to people to work with if we can just do the analysis ourselves and make the results available? So most of these resources fall under the category of genome-wide association study or kind of molecular association results browsers. Main one is this GWAS Catalog, which you can access if you're ever interested in learning about how genes, individual SNPs relate to any phenotype, any disease. This is a good place to go because it doesn't just analyze a single study or a single resource. It cuts across, it looks at metadata from publications. And so you can get sets of publications that are associated with associations of a gene to a phenotype, not just kind of based on one analysis or one sample. But it's also it's gone through growing pains. It used to be that you'd only upload data sets that you only upload genetic associations that are significant at p to the negative 1 times 10 to the negative 5, which is suggestive threshold or kind of lower p-value. And you miss the whole kind of genome-wide landscape in that sense. You can't apply a lot of polygenic methods or genetic correlation analyses with that limited data. But now, as I said, it's gone through growing pains. People are uploading and making available full summary stats. That's really kind of moving forward. The GTEx portal is your go-to for the kind of integrated, the multi-level association of genotype, gene expression, and basically, that's its genotype and gene expression, but in multiple tissues. It's body wide. It's not just in the brain, but it's in multiple regions of the brain as well as multiple tissues and organs. So if you want to know how genes at the RNA level are regulated by genetic variation in some tissue, GTEx is probably the #1 resource in the world to look at. It's an example. You can see how an expression, this SORX3 gene, how is this gene expressed in all of these different tissues that they've looked at. And these are kind of, average, these violin plots showing just what's the level, this transcript per million expression of this gene is across tissues. You see it's highly expressed across many brain tissues. And as I said, it's integrated with genetics, not just where a gene's expressed, it's which genotypes are associated with expression. They're called EQTLs, expression quantitative trait loci. You get a map here of all the SNPs that are associated with genes and also alternative transcripts, splicing of genes within this region. And it's genome-wide, a huge resource. More recently, this FeeWeb, really great browser. There have been a lot of kind of, based on some of the emerging resources of really large sample sizes, every few months there's like a new state-of-the-art GWAS for some trait. I mean, actually, for individual traits, usually every couple of years, 2 or 3 years, but the sample sizes get bigger and bigger. How do you navigate those results? How do you look at them? A paper isn't enough. So there are these, really nice browsers. FeeWeb is one. You can select a kind of cutting-edge GWAS that's been done, select a disease, for example. So here's major depressive disorder, and you can see this is a result actually from the UK Biobank. It's not a clinical sample. And there are reasons why you don't see the same results that I showed on Wednesday when you have a case control study of major depression kind of clinical cases versus controls. This is at the population level. Only 361 cases in this analysis, as you can see. And this is the Manhattan plot. So you can see genetic associations with major depressive disorder, or you can select a gene and a specific SNP in a gene. Here I've selected one of the SNPs that make up the ApoE epsilon 4 allele, which is highly associated with, as you can see, hyperlipidemia, disorders of lipid metabolism, hypercholesterolemia, and dementia. And you can see all of the different diseases associated with the SNP. You can go backwards, right? You pick one disease and see all the SNPs associated with it, or you pick a SNP and see all the diseases. That's like a FeeWAS. The Pan-UK Biobank Consortium, so this is also a kind of results server. This is a set of results that have been made available. Oops. It's a set of analyses that were run on the UK Biobank, which is a mixture of multiple populations, different ancestries. So this is really important for diversity. Obviously, the European sample is the largest of them, the population of the United Kingdom. But they ran analyses on thousands of phenotypes in UK Biobank, full GWAS, full genome-wide associations, and you can get those raw data, the results of those GWAS, and download them from their portal, which looks like this. You can select the population, you can select the description of the phenotype. These are kind of blood levels of certain metabolites and lipids, and you can get the full sets of summary statistics. Here there's over 7,000 phenotypes that are available. And these results can also be browsed, not this exact analysis, but versions of UK Biobank GWAS can be browsed on the FeeWeb that I just showed. In neuroimaging, not going to spend too much time here. We heard about some resources available for analysis. We heard about Human Connectome Project and a lot of other imaging resources last week, so I'm not going to belabor this. I'll mention the Alzheimer's Disease Neuroimaging Initiative, longitudinal, highly detailed, probably the best resource for really in-depth Alzheimer's disease research aside from the Religious Order Study Memory and Aging Project, which is on Synapse. The Philadelphia Neurodevelopmental Cohort, so this is a study of youth, aged 8 to 21, about 10,000 individuals, but it's a cross-sectional study. But again, it's got a matching of, there's genetic data for some of them. Well, they all have genetic data. Some of them have imaging, and they have a large number of questionnaires and kind of additional phenotype data collected on them. Human Connectome Project, we heard about this, kind of just understanding, again, it's a little bit older, but was quite seminal and kind of helped inform our fundamental understanding of structural connectomics, and also can provide an analytical toolbox for like normative brain development. I won't go through this. The NIMH Data Archive. So again, there are these central repositories. This is specific for autism. If you want the NDAR, this is like quite a large group of individuals that have different types of modalities of imaging. You'll see it's actually it's not as large. So you see that there's like 95,000 individuals. They don't all have neuroimaging, but there is a nice kind of developmental data set here with phenotype, genotype, and neuroimaging data all available. It's specific to kind of autism and autism spectrum disorders. Child Mind Institute, again, developmental. So these are kind of developmental imaging data sets. It's very difficult to gather developmental imaging data sets because it's very difficult to recruit and kind of keep young people and their families involved in research, especially really invasive or intense research. And I'll show you an example of a study that we're doing just at the end of this session, which will be in a couple of minutes because I'm going to rush through these resources. My Connectome, Russ Poldrack, what a champion! Out at Stanford, and Aaron's pointing out now everything funded by NIMH is going via the NDA, the NIMH Data Archive. So this is another thing with funding agencies now. Sharing is kind of mandated, despite the fact that the sharing mechanisms are less than ideal. So everything is going to kind of go through there. This is an interesting data set. It's not really a population-based data set. Russ Poldrack just scanned himself twice a week for 18 months. He took blood tests. He kind of tracked his whole life, transcriptomic metabolic data, just turned himself into a study of one. Speaking of digital health and being your own control, looking at how he kind of evolved in fMRI, daily surveys, blood draws over time. And the data set that he kind of derived, that he got out of this, it consists of dozens of different types of MRI modalities, including like 100 fMRI scans. I guess 84 of them were of high quality, as well as RNA sequencing and metabolomics at 48 different time points for one person. So it's just an interesting data set to kind of know about. Open Neuro is a kind of a free platform for analyzing and sharing neuroimaging data. This one is also Aaron is the one to speak about this. There are data sets here. It's kind of there are exceptions to this restricted access. Not all this data is restricted access data. Some of it is provided totally open to play with, essentially, to kind of learn from, because it's not necessarily associated with a lot of in-depth phenotype data. There's not a high risk of reconstruction of kind of an individual identity at the population level. And then PhysioNet, which is another interesting resource if you're interested in modeling of large, kind of integrative signatures, different types of physiological measures over time and in different conditions. It's a really nice resource for looking at machine learning methods and benchmarking machine learning methods. Also, a lot of these data sets are totally open. You don't need to apply through your institution and just kind of access this, and it's a really nice kind of machine learning testing data set. Finally, the major population cohorts. The major population cohorts, these are kind of the areas that I focus on and am most interested in. The All of Us study, which I've mentioned a couple times before, is a really large population-based research study out of the United States funded by the NIH Precision Medicine Initiative, and they're aiming to get to, I think, 1 million people and have the sample kind of be representative of the population of the United States in terms of demographic representations. And it's a really thorough phenotypic collection. So they're basically going for everything. It's like this idea of how many things can you measure, in one person, about lifestyle, about healthcare utilization, medical history. Let's get blood for genetics. Let's get neuroimaging on a whole bunch of them. There's a lot of other studies that kind of branch off from this, and it's going to be a really huge kind of multimodal data set. And as I mentioned, diversity is a really big part of that, and making sure that it's not just recruiting a single population for the sake of homogeneity and identifying effects. CanPath is almost like a version of All of Us for Canada. It's like bringing together a few different data sets that had been collected in different provinces along a similar line. So basically looking at a population level, many different disorders, not selecting for psychiatric illness, not selecting for anything in particular, but trying to capture diversity in the population, get material so we can have DNA, get a large number of assessments for physical health, mental health, and then, within those studies have the opportunity for investigators to launch their own sub-studies to get even higher depth, more invasive types of assessments done. And as you can see, because it's at the population level, it's just not it's not specific to mental illness. It captures a large number of heterogeneous and comorbid conditions that occur generally in the population, including hypertension and arthritis, very, very common, as well as cancer and stroke. And of course, part of this is also collecting information on mental health and mental well-being, and it allows you to look at those things in the wild, so to say. ABCD is another study in youth. The difference between ABCD and the Philadelphia Neurodevelopmental Cohort, the main one is that ABCD is longitudinal. It'll be 10,000 children, and they're going to be kind of followed up for years and years. And that's really important because developmental trajectories are really important, and they're non-linear, and they change as a function of all kinds of genetic and environmental effects. So it's kind of the idea of this ABCD study is to look again at the population level. This isn't just individuals who are affected by mental illness. This is just the general population of young people, and try to identify some normative trajectories of brain function, and then also look at how the variability in brain function and mental health and mental well-being and symptoms of mental illness might change trajectories of development and help us inform models of that. It's quite an intensive study. That's kind of the trend, right? It's something that because we don't have the answers that we want so far, looking at just neuroimaging or just genetics or just our psychometrics or just our environmental assays and kind of social determinants of health questionnaires, we figure you need to kind of know more about each person. You need to put the same person in a scanner, get the same person through a 5-hour set of clinical assessments. You need to give the same person a bunch of push notifications on a phone. I mean, obviously, if they're very, very young, that might not be possible. It's a balance, right? Get more data on more people, but also preserve the health of the people that are participating in the study. It's a very interesting consideration. Finally, for kind of this large scale, UK Biobank, I think it's second to final. UK Biobank is just like all of us, but actually it came first. Probably should have presented it first. This is one that we work with very often. 500,000, so 500,000 people. And again, you have clinical data, genomic data, neuroimaging data on over 40,000 people, population level. And those data sets are growing, and the UK Biobank is kind of making more and more data available. We kind of stand here in 2021, telomere length data for all 500,000 people, metabolomic data for over 100,000 people by the end of this year. And they're moving to a kind of federated research analysis platform at the end of this year and into 2022. So you won't actually be able to download this data anymore. This is another trend. There's so much data now, it's so big that it doesn't make sense for researchers, especially multiple researchers at the same institute, to download terabytes and petabytes of data and try to analyze them on their own and figure it out when you can just log into a server somewhere else on the cloud and analyze it in a protected environment. And that's where they're moving to. FinGen is an example, a very interesting example of a data set. You can apply for data releases. It's actually a bit more constricted in terms of who gets data, but it's another one to be aware of. 218,000 people focused on genetics, but there's a lot of phenotype data made available, and also biobank data because of the Finnish healthcare system and their biobanking that's done. And if you're interested in looking at more data sets, obviously this is just a smattering, a small number of them. Yeah, you can look at this Maelstrom initiative, this research that's been done out of McGill, and this group has compiled metadata from hundreds of studies and consortia, including many of the data sets that I've shown already. And they tell you how many participants are there and what type of data do they have and how many have certain types of data. Very useful. And finally, this is the final kind of 1.5 slides. I just want to draw your attention to a study that is just now launched at CAMH, which is our kind of first contribution to this pool of kind of larger growing, major cohort studies. We are aiming to recruit 3,000 youth between the ages of 11 and 24 and follow them for 5 years. And again, as is kind of really important for this integrative multi-domain research, we are collecting a really wide range of biosample information, brain scans, a really detailed set of assessments for neurocognition and educational attainment, and also merging with Ontario data for healthcare service utilization, using OHIP and this kind of available governmental agency to extract interactions that every person has had with healthcare outside of our hospital. And we're going to address multiple aspects of youth development. The key difference between this study and something like PNC and ABCD, other kind of large cohort studies in younger people, is that we are actually only recruiting individuals who have a psychiatric diagnosis, who are coming through the hospital system and are seeking treatment for a mental illness. And that's a very interesting population that's understudied because it's very hard to kind of access that population, and it's always observed, it's been seen in other studies that people, young people with a diagnosis of any kind of mental health disorder in youth, they're really early age, are more likely to develop other mental illnesses later in life. But yet that population remains unstudied longitudinally at this level, and many of our investigators at KCNI are leading different aspects of this study, including myself for trajectories of whole person networks. Aaron, Dickey, and you heard from Andrea and John are all participating in the imaging and molecular profiles side. So that's kind of compressed version of those. So I hope at least one of those might have resonated with you. If there are any questions you have about a particular resource, just put it in the questions, or I'll be in Gathertown for a bit. Just don't worry about it. Just ask me about it. You can ask me on Slack. We can talk about it, and I can kind of help you out. I'm going to hand it off to Abhi, who's going to take us home in the next 10 minutes. We're pushing it. So I'll skip through. We're just gonna redo Joanna's. I hope no one's hungry, because we're not going to do lunch at all today because you haven't had enough science. So we're going to jump through. Abhi, would you like me to go through the slides for you, or do you want to share them on your computer? Sorry, I didn't get that. It was a little bit garbled.
You can
okay, I'll go through them.
Yeah.
Okay.
Then you have to Okay. Hopefully everyone can hear me. That was crazy to hear myself back over. Okay, I'm between you guys and your lunch and my morning coffee, so I'll make it quick. And here is a complimentary sort of view, and actually the setting worked out very nicely. So Dan has clearly laid out there is tremendous amounts of data available. I think there is no doubting that, hopefully. There are other concerns, and I'm going to talk about that, but hopefully it's very clear across the spectrum of different kinds of data that you might be interested in. There is one or the other data set broadly available after some governance access issues and all that. But where do you sort of bring it all together is, next slide. Oh, it's coming out weird. It doesn't matter. So the point is lots of data. Next slide. But that also in itself is an issue, right? Where is the context coming from for this data? If you open neuroimaging, openneuro.org, or any other sort of repo that Dan talked about, it becomes a little messy to understand what do I download, where, how do I contextualize this, what am I comparing with each other, what's my outcome, what's my response. So I'm going to talk about that for very few minutes. And the slide that you have here on the left-hand side shows a sort of a typical sort of a workflow. In this case, it's from UK Biobank. That is multimodal temporal data collected from a large swath of population. But as a researcher, and probably as a young researcher focused on various different interest areas, you typically can get overwhelmed very frequently. At least I still do. I mean, I'm like, what are we trying to do? What's the main outcome of interest? What's the confounding variable? So the point is, raw data, whether it's sensor level data or genotypic data, is not enough all the time. The underlying context in what are you trying to analyze it for is actually super important, and some philosophical thinking on that side before you get to coding and downloading and all that stuff, I think is really helpful. So while it's not the most exciting time to think through and write that, but I think that's the most important aspect of it, and hopefully can help with the fastening the analytical timeline. So few factors is, what are you trying to analyze? Are you trying to combine individual level data? In this case, possible for UK Biobank, because the same individual has been phenotyped digitally biologically. That's great. Certain times, that's not an option. You're trying to integrate a cross study. Will you integrate by socio-demographic factors, or what? How will you bring someone's external environment into play? So these measures are collected both in clinic and at home. We talked about that earlier today, right? And so there is variability and different amounts of variability. Next slide. From where these data are collected, from whom, when, how long, under what mechanism, under what conditions. So my take home on this is, it is super helpful. I'm going to state the obvious, but spend some time reading through the underlying basic data characterization papers. That will help you understand from the data generator's perspective. Important to highlight, right? People who produce the data, on what are the underlying assumptions or factors which were responsible for data generation. Sometimes we ignore that and get to the data right away, and we miss the important pieces there. So that can bring in a lot of context, and as well as introduce you to, these were from northern part of the England or southern part, and there is differences in environment that needs to be accounted for. Next slide. So the context matters, right? I'm recycling this slide. And the more you read about that data set, under what circumstances it was generated, from whom, when, etc., will help you get a better hang of it. That can inform your hypotheses and external data sets that you might need. Next slide. And it also makes a whole lot of difference what kind of data set you're trying to analyze. As I mentioned, traditional health research sort of data in clinic collected, going very fast. And then remote mobile-based digital health research data collected remotely. They all brings their pros and cons, and we have talked about that. There's one more slide that hopefully we'll get into it. Next one. Yes, so for example, I'm trying to assess someone's gait and balance in Parkinson's or any neurological disease. You would expect that exercise to be conducted, in this case, through a smartphone, and you're measuring gyro or axle data. So these are onboard sensors that are available ubiquitously across smartphone devices nowadays. And you would expect some shaking XYZ on an XYZ axis. So this is a tri-axle data. So XYZ axis for an accelerometer that's on a smartphone. But in some cases, we see a flat line. Are people not completing that task? Are people doing that task in a wrong way? If you don't sort of look at that QA/QC of the data and just mush it all together into your machine learning or statistical analysis pipeline, you will average out or nullify the effect in some cases. Not at all. Depends on the proportionality of missingness or noisiness. But that exploratory analysis of what is the raw underlying data, how it should behave and how it is behaving based on what you see, is super important. And that's reading between the lines. This is where the data science meets the art, right? It's the contextualization of what you are trying to go after. And simply put, if you see a flat line or uniform distribution for axle data, something is clearly wrong. We need to do something about it. And machine learning will not tell you until you sort of understand the context. Next slide. Oh boy, we might take some more time than I expected. So here is an example of external data that is super useful in my research in digital health. So the health outcomes are related to health factors that are related to health behavior, clinical care, social and economic factors, physical environment, you name it, right? And it's hard to sort of say, I'm going to combine all this data, find all this data, and sort of make sense of it, when some of this work has been done beautifully by other funders or other groups, right? So one of that is Robert with Johnson County Health Ranking data. Admittedly, this is US only, but I have a feeling that at least similar things exist in Canada, or at least could be. And so here's that example, right? So you can actually let's look at mental health, right? Social and economic factors. Let's see, I'm going to focus on quality of life. Poor mental health days, you can click on this. It will take you to various different contexts on where this data came from, etc. You can explore the data. Let's look at Washington State, my home state. And here is the data, right? So different counties in Washington and what's their sort of average value across the board, error margin. And this is super useful data if you are doing some geospatial contextual analysis to look at county level differences across your wide swath of data in the future, right? So it's available, it's downloadable, very nicely cataloged, and something to remember as you sort of bring it together. And I just realized I was talking, thinking that I was sharing the screen, and Dan is sharing the screen. So my bad. I just went on my track there. Dan, maybe you can click the demo too here. So this is another amazing resource, demo too. It's not covered.
I was just doing it. I don't know if anyone else saw it, but I had it. Whoops. I did have the demo 2 open. I was trying to show it on the screen. So okay. While you were talking, I was doing this.
Thank you.
Sure.
Okay. People have then can you do that? Okay. Sorry, this was intentionally for the screen sharing on my side, but anyways, the point is there is these 2 data resources available in the US context at least that should have Canadian counterparts at least at the data level that could be used for finding data, shopping for it. And the demo 2 basically talks about how can you shop for data, create a data cart, and download it. I have used it many times in my research for contextualizing data. Next slide. And this is I guess the short summary of this is, as you are planning your modeling exercise, we all think about what's our response or outcome, whether it's depression severity or whatnot, right? Anything that is of interest, and what are the covariates, right? Person-specific external factors. This is where it all needs to come together. You need to think of what is my unit of analysis? Is it person? Then what is the person-specific context? What are the external factors that are of interest to the study, and how can I bring that together? And then what are the neighborhood factors that are statics of sorts, because neighborhood characteristics don't change daily or monthly, and how would you join those together? So it's not just that person or that cohort and here is an outcome. Remember, all of this is impacted by our lived experience in the world we all experience, live daily. So it's not just the baseline study characteristics that's important. They are, in fact, being shaped by our local environment. So factoring that in is super important, especially in mental health and especially in low resource settings. There is a whole day and night difference in what might be a mental health issue in Orange County, California versus Maryland, predominantly Black zip codes. And it is what it is in that case. Next slide. Next one. Here is another example. Dan talked about UK Biobank. You can contextualize that data from the social environmental factors using a very awesome package called Fingertips R, which basically provides a direct interface to the UK social environmental and health data stats. And here is an example. You can download it very quickly, look at various health improvement, health protection metrics, and then find the unit of analysis. That's important, the zip code, postal code, or city, etc., and then do that join and contextualize that even more. Next slide. Dan has talked about Synapse much better than I can as an evangelist. Thank you, Dan. Sage would have been happy. So Synapse has another portal which is on new and upcoming. This is specifically focused on digital health. And so not going to do a demo on this right now. It's very similar to the other genomic data portal that Dan showed, but it's a very good resource and growing resource for finding individual level, sensor level data. Those are the keywords. It's very difficult to find sensor level data for individuals in digital health because of the privacy issues. So you can find it there. You'll have to prove your credentials and context and all that in terms of your academic affiliation and all because of privacy issues, but it's available. Next slide. So this is where the transition will happen in the next session. So we talked about many sources of digital health data, the data deluge. Dan showed you so many examples. Joanna showed you a clinical example in a clinical setting and the context that I talked about. But the insights that we draw from this analysis, how robust are they? Can they stand the test of a second researcher looking at your paper and reproduce that? So I'm going to talk about that in the next session and doing some hands-on analysis or a demo, depending on how many of you want to follow along. Dan, is there a next slide?
Negative. There's not a next slide.
Okay. So I am probably it's on my side. It doesn't matter. Once we get started, and if you did get the instructions from Erin, I'm not sure. I'm going to spend 10 minutes of that session to give you some time. Let me. So you guys have an opportunity to create an account and then hopefully get certified. That might not be easy in a few minutes, but rest assured, my demo and the talk will be about the concepts of reproducible analyses and not just focused on a tool. So more on that in the next hour after break. With that, I'm going to stop here.
Thanks everyone. I just wanted to also point out there was one question that was asked by Akshay, and Akshay, we will get to that question in the afternoon session when we have some more time. We'll let everybody kind of get to the lunch. Thanks everybody.